Hello everyone, my name is Kimberly Cranmore and I am an undergraduate at the University of Central Florida and I am studying biology. And this summer I worked in Dr. Jamie Foster's lab and, well, I worked on the project. It's a big difference from everyone else's projects, but I was working on the genome sequencing bacterium in modern lithifying thrombolites. So what are thrombolites? Well, basically they are rock-like structures that are made from the metabolic activities of microbes. And a microbe that is most significantly dominant in the thrombolites are cyanobacteria. And the cyanobacteria are actually able to precipitate out carbon. They precipitate out carbon as calcium carbonate, and so the thrombolites are actually rock-like structures that have a clotted formation. And why are they important? They just look like rocks. They actually represent one of the earliest microbial ecosystems in the world. The oldest is actually stromatolites, but they are both believed to have created or produced the oxygen that was first on Earth and was used to actually sustain and allow life to actually be possible on Earth. And so because of that factor, we can actually use these thrombolites as a model for understanding the evolution of life on Earth. One of the best studied thrombolite ecosystems is located in the Bahamas at Highborne Cay, and this is actually where we collected our samples for this project that we looked at. And in Highborne Cay, the thrombolites are about 10 feet deep in the ocean. Actually, Jamie and her team had to dive and collect the samples. The thrombolites are protected under the water by the sand. They're essentially covered in sand. So, Dichothrix. This is the main species that we're looking at in this project, and it's a dominant component of thrombolites. And it's a bacteria that is in the thrombolites, or we believe is a major cyanobacterium in the thrombolites that were in Highborne Cay. Dichothrix was the species that was focused on because it was, again, the dominant cyanobacteria. And since it's a cyanobacteria, it goes through photosynthesis, is able to take out calcium carbonate, and so we want to better understand how it's able to do that. And so we can do this by sequencing the genome of the Dichothrix, which is the dominant organism in the Bahamian thrombolites, we believe. So we actually could not cultivate the exact Dichothrix. So with our data, we were able to create a mini so we could improve the recovery of the Dichothrix. We think this is because the Dichothrix is such a small organism, and we had to dissect it out, and it had, well, there's actually a good amount of contaminants, so you'll see that. This is actually the Dichothrix. This is what it looks like. It's about the size of half an eyelash. And so our job of dissecting about 2, maybe 4 grams total of this Dichothrix. And I did this using a microscope, or dissection microscope. And as you can see, it's pretty filamented. I used an average of 4 times magnification while I was dissecting. You can see there's a lot of, it's called them ooids, and around here there's lots of sand. I had to pick that off. But yeah, that's what Dichothrix is. It's a cyanobacteria. It's sort of green in color. Dichothrix the DNA was extracted using a xanthogenate method, and I was able to improve this protocol by using liquid nitrogen to break open the cells of the Dichothrix. It's such a small specimen, I guess you could say, and there's just so few cells. So we really had to break them open, and thanks to Rochelle with her big tubes. After we collected extracted the DNA, I used a Zymo clean and concentrator kit for genomic DNA, and finally, after weeks, I was able to collect enough DNA for sequencing, and in total, I collected 675 nanograms of Dichothrix DNA, and that was in a volume of 25 microliters. And then it was sent out for sequencing. We sent it to the University of Central Florida.
No.
They had, or University of Florida. I'm used to that. The University of Florida. Sorry, my bad. There, they used the aluminum biseq reagent kit B3, and this was able to read base pairs at 2 times 300. And so far, we've been able to put we've gotten our sequencing data back. And so far, we got in total around 20 gigabytes of data. And we've only been able to analyze 1 gigabyte. We did this using MGRAST, which is a database that allows you to assemble and analyze metagenomes or genomes. In the future, the near future, we will use Velvet and MetaMos to further analyze, assemble, and the genome, metagenome now. And that's a picture of the aluminum biseq. So, again, this is 1 gigabyte of the data that I have collected, or that we have sequenced so far. This is how many reads we had. That's 4.5 million reads from only 1 gigabyte of data, which is crazy. Source and 5NR is just another database that they looked at, and they were able to get this data and compare. So we did see cyanobacteria in this small bit of data. Unclassified is what we really want to look for, because Dichothrix is unclassified in the cyanobacteria. It's not sequenced yet. And so we saw an abundance of 869,000 out of this 4 million, which isn't great, but at least we saw a good amount. And 77% was identified, and we had 69,000 hits in GenBank. All right, so this is the genome chart. It's just an overview of the sequencing data. don't know if you can see in the middle, but that's the 4 million reads. So in this area, we see a lot of streptophyta, which we don't want to see. And what we're looking for, if you want to compare, is this cyanobacteria. That's a small bit of the, I guess this is kind of like a more complex pie chart. And over here you see the proteobacteria, which is also really significant. The streptophyta is, it represents plants, I think. In this case, it's probably algae. And algae does grow off of the Dichothrix, and any bit of the algae that was dissected or got contaminated while dissecting could definitely throw off sequence. And then you see over here eukaryota. In this area, it's also pretty large. There's a lot of species that was seen. But if you do look at these bars right here, you can see that they're higher than a lot of these bars and a lot of these red bars. And that's a good thing because they're more abundant, I guess you could say. Closer look, again, you can, a significant abundance of reads for this cyanobacteria area. And a bad thing though was this red area, which was proteobacteria. Proteobacteria is E. coli, Salmonella, Vibrio, those kind of things. So that wasn't too good for our findings. Future directions. Of course, we have lots more data to be analyzed. This was only a small snippet and it was not randomly selected. So 20 gigabytes of data to be analyzed left. Hopefully that will happen soon. And of course, again, the possible source of contamination during dissection. It really is, this is how it looks under the microscope while you're dissecting. And any bit of extra stuff really can throw it off. You can see these, like I said, cyanobacteria and Dichothrix precipitate calcium carbonate. You can see the kind of little, they look like sand, but they're calcium carbonate. Again, the streptophyta and proteobacteria, there was a lot of that. So, the implications, again, we want to figure out how Dichothrix, or what genes actually precipitate carbon in Dichothrix, so we can better understand how the metabolic activities of microbial mats, and because they are one of the oldest microbial ecosystems in the world, and they did produce a lot of oxygen when the world was first coming together. And this will be again the first step in assessing the metabolic complexity, which is what we really are hoping to do soon. And this project, I'm actually going to present it at the ASM meeting, the Association for the Society of Microbiology in Ponte Vedra, Florida in September. So I get to present a poster there with Diana. So that's a good opportunity. And I would like to acknowledge Dr. Jamie Foster for allowing me to work in her lab this summer, and my graduate mentor, Diana, and of course everyone in my lab, and Space Florida and NASA Florida Space Grant Consortium for the opportunity. And thank you. Any questions?
Yes, can you go back to the picture of the filaments when you're dissecting them?
Which one? This one? Or?
Go back a little bit more. Yeah, what's the whitish, like, transparent substance, like the gel-like?
What's the question?
What's the gel-like substance that's surrounding the filaments?
That's seawater. Or actually, no. the samples were preserved in
Talking about the carbohydrate, I think he's talking about
carbohydrate.
the exopolysaccharide stuff, the gooey stuff.
Oh, the gooey
Yeah,
stuff.
yeah. All right.
Yeah, there's a lot of junk in, in the samples. Yeah, like
Did you try cleaning the samples at all before grinding them in liquid nitrogen? I don't know what.
I did. I actually used the which was it? The RNase-free water right before. Because actually when I put the samples after I dissected them in the tube and I put them in the freezer and I preserved them in RNase, they actually precipitated out like salty substance. It was really weird. I think it was from the salt water that was in the actual sample. So I had to use the RNase-free water and kind of shake it up, vortex it, and then I took that out, and I don't know. There's still a lot of contamination, but oops. So, yeah. Thank you.
So, Kimberly, is that your first?
Can you guys, yeah.
Sure.
Can we have